Hello, all. A warm welcome to the podcast series being introduced by the Center for Brain and Mind here in Bangalore. Today, I am joined by Professor Jens Hjerling-Leffler, who's a professor of molecular psychiatry at the Department of Medical Biochemistry and Biophysics at the Karolinska Institute in Stockholm, Sweden. Welcome, Professor.
Thank you.
So happy to host you in Bangalore and have you in India. We'd like to first know about your journey into the field of neurosciences and how is it that psychiatric disorders became one of your focal areas of studies?
Yeah, well, why I started out as a what we call in Sweden a green biologist, so ecology, and I really like bird watching, and I wanted to work in the field. But then one day we had a system that you had to, when you applied for a course, it was competitive to get into the different courses, and in the morning you had to be there when the course started to claim your spot, and you had to say, I'm here. And one morning I overslept, and I missed the intro to Ecology 2, and the organizer called me saying, where are you? And I was like, I'm sleeping. And then because I couldn't take that course, that my spot went to someone else, I got into the neuroscience course. And then I took the neuroscience course, and that's when I realized that this is the coolest thing there is. So then after that, I was still in the weighing back and forth, whether I wanted to spend a life outdoors in nature or working in a lab. But then I realized that neuroscience can only be done in a lab, or and this type of neuroscience, and I can be out in the nature on my free time. So then I chose a career in neuroscience, yeah, I never looked back. I really love neuroscience, so it's
interesting. So that was an accidental entry into the field of neuroscience.
Then I started with electrophysiology, which I think is super interesting. Really a place I love working with my hands and getting. You see the data as it's coming, which I really like. And then I started out as an electrophysiologist. But then after a while I realized that to me it was a very good readout of the function of a neuron and so on. But I felt that if we want to ask good questions, we need to manipulate the system somehow. And I felt that at the time, there started to come this thing, the knockout mice, so you could manipulate gene expression, and I thought that that's probably how I want to do it. I want to be able to manipulate gene expression and change the phenotype of animals, and then use electrophysiology as a readout. So that was my starting point. But at that point, I didn't know any molecular biology, so I then went and I did a PhD in a molecular biology lab. And turns out I'm really bad at molecular biology. I struggled, and my PhD was kind of tough until I started doing calcium imaging. So it's, again, back to the physiology, which I really like. But during my PhD, I had learned a lot about molecular biology, and I realized that there was these new kinds of mouse genetics that came online. Then it was in 2005 or so, where we could, using this Krelok system and fate mapping, we could label cell types very accurately. And that was the first time that I felt like it was time to move to the brain, because before that I worked in the peripheral nervous system. But I realized that if we can label specific cell types with fluorescence, then we can kind of go back and record from the same cells, because electrophysiology before that had been you stick a needle in and you patch onto something and you didn't know what it was. You just hoped it was the right cell. But now we instead had these fluorescent cells, so we could start looking at these rarer populations and be sure that we recorded from the same cell over and over again. And that's when I felt like this was the big piece that was missing in order to really understand the biology of the central nervous system. So that's when I started my postdoc at NYU with Gord Fischel, because he was really a pioneer in using these kind of mouse genetics in order to understand brain development. So I worked there, and the main thing that I set out to do is try to understand what is a brain cell type, where does it, it can be, you can classify them in many different ways. It could be morphology or firing patterns and so on. But with the tools that was available there at NYU, we could actually classify cells on where they arose, where they came from during development. So I spent a long time doing that, and I think me and my colleagues, we together figured out kind of like the cellular diversity of an important set of cortical interneurons that derive from the caudal ganglionic eminence. And so I was very much into trying to understand how many different cell types are there, what do the different cell types do, what kind of computation do they contribute to in the circuit. But I really liked development as a way to look at function in the nervous system. So I was awarded a very position from the Swedish government to come back and start my own lab. And when I was interviewing for different positions, I met this guy called Sten Linnarsson at the Department of Medical Biochemistry and Biophysics. And he had just discovered a way to do something called single-cell sequencing. And he had done it in cell lines. And I was really blown away because I felt like this is the way to do it. So that made me, and other reasons too, but I accepted this position at this place. I went back to New York and spent a long time trying to figure out how to dissociate individual cells from the brain, because that was something that was not so. You could dissociate cell tissues. That had been done by a lot by developmental biologists, and during the embryonic development, cells are very robust, so you can just do anything to the embryo and the cells are still happy. But what we couldn't do at the time was to do that with adult mouse tissue. So me and a grad student at NYU, Tricia McKenzie, we worked to do this and solved it. And I think that was the key, because then when I started my lab at Karolinska, me and Sten could start collaborating because I could do the dissociation of the cells and he had the chemistry to do the single-cell sequencing. And that led to, I think, the big breakthrough paper when we, for the first time, mapped molecular cell types in the mouse brain. And I started my lab under the premise that I wanted to understand postnatal brain development and adolescent brain development. And the reason for that is that we had some data showing that the same genes that are active during embryonic development, they have one role there, but then if you perturb them a little bit later in development, they did something completely different. And these are transcription factors that are thought, not anymore, but at the time they were thought to do a thing. Like, this is the transcription factor for this cell type for this. But it turned out that during the time of even if we just switched the temporal window with like 1 day, they did something completely different. So then the idea of my lab was to develop tools that allowed us to manipulate these transcription factors, or these kind of, to see what they did in adolescent brain development. So then I worked on that, and I got sidetracked by this single cell work a little bit, because that took off so much. So we did a lot of work on single cell biology. But now we're back. And so I'm a basic scientist. I was always a basic scientist. But then I kind of got fed up with writing research grants where I used disease as a way to get money rather than being actually interested in the disease. And then so I decided that, and this is where two streams kind of converge. One is my biological interest in adolescent brain development. And then there's, in my family, we have a history of mental illness. So my aunt has been hospitalized her whole life. And so I felt that there was also like a personal, deeper connection to wanting to understand these disorders. So that's when I decided that, and I had a brilliant postdoc in the lab, Nathan Skeen. He had a really great idea to see if we could marry the single cell transcriptomic data to genetics of disease. And I've always was really interested in genetics, but I never really learned how to work with it practically. But this was the chance, and the idea was really great. And I said, okay, but if we do this, we have to do it properly. So we should collaborate with the best geneticists. And I was lucky to get to know Patrick Sullivan at UNC. He had accepted a professorship at the Karolinska at the time. And so I started working with him, and that was the beginning of a very, very long, now it's more than 10 years that we've been working together. He taught me genetics. So now I do genetic research, but before that I didn't really understand what a SNP or a GWAS was.
So there have been interesting people coming into your academic journey and
yes,
kind of training you, teaching you.
I think this is what science, I always say that it's good to listen to podcasts, but the real science happens in the meeting between people, I think. I think it's good to meet in reality. And I think also, science is an amazing career for many people, and I think the main reason is that you can really turn it into your own. So I'm a person who is social, I like to talk, and I like to connect to people. So to me, that's been my scientific. But you can also be someone who likes to sit alone and think. And I think in science you should play your strengths. So this is my strength, is to meet people and working with brilliant people is fun.
So you briefly touched upon the postnatal and adolescent brain development. So what critical changes do you think must be occurring at these stages, and do they really reflect in the way an adult brain develops, and in the adulthood, is there any manifestation of it later?
It's a good question. I think that it's a separate period. I think it's evolutionary conserved, so almost all species goes through this, especially like at least mammals. And I think it has a purpose. The purpose is not to build an adult brain. It's a purpose, so this is the years when you are so I'm talking about kind of late adolescence, so not early adolescence. And adolescence is a bad word because it spans such a wide window. So it spans from puberty all the way to adulthood. And I kind of think that, puberty, you're still a child. So I'm interested in this late adolescent period, maybe when you're, 17, 18, and until, my dad said you're not an adult until you're 30. I kind of tend to agree. I don't know. It's probably different for different people. So in this period, there is a number of physiological and behavioral changes that I think are very adaptive to. One is that this is at your peak of physical strength, and also a lot of the cognitive abilities peak at 19, 20. And at this time point, you see behavioral changes, and now I'm talking not just about humans, but also about mice, is that you turn away from your family group. You're now suddenly much more interested in pay attention to outside your family. You take a lot of risks. You move. You also need to eat more, and you sleep longer in the morning. But I think it's a period that is necessary for us to mix, to meet new people, and to not just stay in one place. And so this is, I think that it's kind of like an important period of its own. And then brain-wise, I think that it's pitifully understudied. I think like we don't know, people tend to overlook it. The two things that people say is that there's synaptic pruning and increased myelination of the PFC. That's it. And if you go to a textbook, that's pretty much what it says. And so I think that there's a lot of fundamental science to be found out what happens in this time period. And for psychiatry, I think that most people, everyone has to make a bet. We don't understand psychiatry. We don't. So you have to make a bet on what you think is interesting, and then you go for that. And you should be careful to speak badly of other people's bets until you've proven your own. So I think that. But the bet that I've made is that I think that the genetic changes happening and not just genetic, but genetic and physiological changes happening during this time period are vulnerable in psychiatric with the genetic background and I think that there's good evidence for this. That's not to say that I think something as complicated as psychiatric disorders, there is also an early development component. And for things like ADHD and autism, that's the main thing, right? Something like schizophrenia, bipolar disorder that I'm interested in, I think that there's an early developmental component too. I just don't think it's my bet is that it's smaller than what is happening later. So that's the bet. Yeah, there's a lot of reasons to why I made that bet. But I think that it came back to this idea that, so if you take transcriptomic, if you take the brains of schizophrenic people when they're adults and compare them controls, you get a signal that says neurodevelopment. So some people think that that means that something happened in the embryo. I think that the word neurodevelopment is probably wrong because it means that these processes are happening in the adult. And they're probably not development anymore. They are plasticity or some kind of adaptive program that is playing out in the adult. And so now it ties back to my idea that these transcription factors that are important early does something else later. So that's still the bet, right? That these transcription factors are still active during adolescence, but now they're controlling different kind of programs. And so that's what we're going after.
So, which is why the late adolescence period, which you were particularly keenly working on, is possibly one of the areas where there should be more focus on, and maybe it's the bet.
I think so. I think I'm happy to see that it's actually happening. So 10 years ago, there was very few research groups working on this, but now there's more and more. So I think that's good. It's good to have a field around you. You're not working on it alone.
So who knows, maybe there's a parallel narrative coming about not naming everything and linking everything to the embryonic reasons. It could be some other stages of the brain development.
Absolutely. And I think postnatal period is very interesting. Like the early postnatal period is very interesting too. There's good animal model work showing that is very key to your final phenotype. And so I think we'll figure out that different things are being programmed at different time points, and I think that's definitely the case.
Correct. So are there any other groups around the world who go by this argument that it could be it could be in the late adolescence along with you?
Yes,
are there other believers?
I'm not going to name them because I don't want to out them.
Sorry,
but yeah, there's increasing, and some of them have been working on a long time. But I think the kind of disciplines that have been working on this, imaging field has been on this for a while. Some electrophysiology, EEG people have been working on this. Behavioral scientists have been working on this. I think it's very clear. What I've been lacking is the attention of the geneticist and the mouse neuroscience crowd, which I represent, that's my background. So I think that's where the big hole is. Imaging, Paul just showed amazing data on how the brain changes over the adolescent, and that's why we have this thing of like myelination and synaptic pruning. That is to a large extent imaging data that it's based on, but the mechanisms behind it. So the questions we're asking now is like, is it specific circuits? We don't know. Is it what causes it? Is it intrinsically programmed in the brain, or is it environmental factors, or one of my favorite questions that I don't have an answer to is like, does a neuron know how old it is? Is there like an intrinsic clock? And there is an intrinsic clock on a daily basis, right? But how long time periods can it keep track of? Does it know that it's 18 years old? Or that's really fascinating.
How much self-aware is a neuron?
Yeah, and because adolescence is remarkably conserved. It happens in everyone. It happens in almost every animal. It would be weird if it's not programmed. I don't think it's, and it's not. It doesn't seem to be cultural, because and I don't think it's linked to. My first idea was that this was linked to puberty, the sex hormones. But actually now there's data in parts of the world that are with increased nutrition and maybe some chemical interference. Puberty is moved earlier.
Yes, yes.
By several years, but brain development hasn't. So I think that that's an argument to actually decouple the two. I don't think that brain development is, like some aspects are, but this general adolescent program is probably not triggered by sexual maturation, I think, because that has not moved earlier.
So the brain is following its standard timetable.
Yes,
whereas
to a large extent, yeah, and the
moved
body
ahead.
is doing something else. Yeah. Yeah.
Another area of your research is schizophrenia. Would you like to talk about it? And where do we stand really in providing treatment for schizophrenia?
Well, I can talk about schizophrenia at length. I think it's a very intriguing disorder. First of all, it's a massive health problem, and the treatments that we have are less than optimal. I mean, there's a lot of side effects. Some people don't respond. We mainly treat the positive symptoms. So I think from a scientific perspective, schizophrenia is really interesting because it's kind of like asking the question, how does the brain work? What is it to be human from a philosophical perspective? What is your mind, when your mind changes, do you change as a person or not? I think these are all really fascinating questions. But I'm not a clinician, I'm not treating people, I should say that. So development, happened by happenstance, right? Like the current treatment was found by a chance, and we refined it over the years, but it's mainly been the work of pharmacologists rather than neuroscientists. So neuroscience has contributed very little. And I think neuroscience has to change, and that's the idea that we work for. Is like traditional neuroscience. I can't do regular neuroscience because then it's not schizophrenia. I can do great neuroscience and find principles, but they're probably not that disease related.
So it's been building over somebody else's work, but as you said, neuroscientists have really not made much as a contribution to the.
No. There are new. There's like, I don't know the timetable, but like recently there was a new drug just approved, and that's a huge leap forward because it's the first time that we have something that works at a different principle. But I think that developing treatment. So my program or my idea is, I don't think I will be able to develop treatments. I've spoken to leaders from the pharmaceutical field, and they agree on that. I think my goal is to bring them back because the large pharmaceutical companies has left psychiatry.
Yeah.
They don't do
So
psychiatry research.
before you proceed, I would like to know why did they leave, and why is that the private players or pharmaceutical companies no longer willing to partner?
Yeah,
What is
exactly.
what was
So
the problem?
I actually met with them and a few of them and asked them this, and what they pretty much all said that there's two main reasons. One is that we don't understand the biology, so they don't know how to pick a target. And the second is that clinical trials are really bad because there's such a heterogeneous response to the target that it's very hard to find a good effect. So they need two things. One is that we're trying to understand the biology, which is something that we're actively working on, right, as a field. But the other one is that we need to understand the heterogeneity. We need to have ways of clustering patients, so you can test a drug in this set of patients, or in this set of patients, or in this set of patients. Because then if you treat them individually, then you have a chance of having success in maybe one of them. So if it is different, indeed different diseases and not just one disorder, then having this patient stratification is going to be very important. And I'm very hopeful because there are now, there's some amazing preliminary data, or first papers are coming out.
Sure.
We heard today about something that there's maybe 2 different imaging modalities where the cortical thinning starts. There's work on the genetic data showing that there are different subtypes. And so I think that we're starting to see that there is something there, and I hope that we can figure that out over the next
So maybe
years.
it'll be the other way around, the neuroscientists going with their research ideas and research results to the companies and maybe have them back on board.
Yes. So oh, there's the big spider. They promised me that they would come back if we solve these problems.
Yes. But do you find it interesting to be engaging with that kind of the stakeholder, because generally scientists work in the lab, as you said, you don't get many opportunities to. How has that engagement been?
I think it's gotten me more grant money, which is the one thing that scientists care about the most. I think that I think we heard yesterday or 2 days ago in the Manatsova that we have to do this together as a society, and I think it's important as a scientist. As a scientist, you're faced with, not daily, but very often you have to make decisions. Do I research this or that? Do you go for this question or that question? And so it's a very target-rich environment. You have to pick your research line, and you kind of evaluate that as you go. And I think that it's important for scientists to think about why they are doing their science. And when you then pick something, you can figure out, so this was a part of my process in order to figure out what are the kind of questions I should be asking in order to have a larger impact.
Right. What is your take on international collaborations, and now that the world is, it's interconnected, shrinking, or what is your take on the collaborations, and what more can be done or needs to be done?
I think that scientists should be left alone. They should be given money and not be meddled with. Because science is truly an international effort, and it has always been. I think that the important thing with it's something that binds us together across cultures, and it's the curiosity of trying to understand how things work. And therefore, I think it's how we divide up fame and money and so on has gotten in a bit of the way. But I think fundamentally in science, there is this wish to know how things work. And when I read a paper that has some incredibly new finding, I get so excited and happy. Sometimes I feel like, oh no, that was my idea. But in generally, it's great, because then you can move on, and then you can have so I think that it truly binds us together, and it's very it's a liberating feel to come to India and meet people that ask the same questions, very different backgrounds, but we still ask the same questions, and we have slightly different ways of looking at life and looking at and I think that the cross-pollination of different perspectives is really important. And to me, I think that's one of the most important things that a young scientist can do, is to travel the world and gather around different perspectives. I think it's good to move, do your master's here, do your PhD somewhere else, do your postdoc, then get to understand the global scene, and I think that's really important. And then that's like during your development. And fundamentally, but I also lead this international consortium, right? So
yes, precisely. I was coming to that, and I would like to know about the Psychiatric Genomic Consortium.
Yes.
And anyway, you see India coming into the consortium a little later, or
No, I'm here to recruit. No, I think so. The Psychiatric Genome Consortium has been around for many years, and it was in the same way that the Enigma Consortium. It was born out of the frustration that people were having this amazing technological abilities to assay how the genome differs between patient and controls, etc. But they were done in 2 small cohorts with not enough and with not the same methods, and so there was a need to coordinate it, and that's been a huge success. We really understand disease genetics much better now. So the group that I lead is part of the PGC, which is called Working Group for Functional Genomics, and it's kind of like the post-GWAS. The rest of the PGC is really trying to understand the disease genetics. It's sometimes said that we live in the post-GWAS era because the genetics have given us so much insight, like so much data and knowledge, but we haven't really translated it into biology yet. And this is kind of like the idea is to take this genetic data, try to understand in all these disease variants that all contribute a little bit. In which cell type do they act? When during development do they have their action? And so genes are specific, but it turns out that these regulatory elements that lie around the genes are even more specific. So a single gene can have 10, 15 regulatory elements. And we heard today that one gene is driven by one regulatory element in astrocytes and another regulatory element in neurons. And so it means that the gene is specific to both astrocytes and neurons, but the regulatory elements are actually specific to each separately. So they are more specific in many ways. So trying to understand how the disease genetics affect these kind of processes, I think, has great hope to making us understand a little bit more what the genetics means. But there are many hurdles, and one of them is again, there's right now we're in the stage of the field where people are doing this on their own in their own lab and reporting it. I think we need to do this we need to understand how does different experimental conditions affect the results, So we're doing a lot of benchmarking or trying to do standardization, optimization of these techniques in order to try to bring us to the same success that the PGC and the Enigma has had in terms of standardizing procedures